This is the lecture for Chapter 20, Antimicrobial Drugs, Part 2. The first part of the lecture went over antibiotics, and antibiotics are antimicrobial drugs that are selectively toxic to bacteria, to prokaryotic cells. Also with the antibiotics, I discussed the major modes of action that they can have. In other words, the different parts of the prokaryotic cell that they can target. So the first target is the cell wall. Second target is the ribosome, which performs protein synthesis, in other words, translation. The third target can inhibit the enzymes that affect nucleic acid replication. So this can inhibit transcription or DNA replication. Fourth target is the plasma membrane. And the fifth target are enzymes that catalyze important metabolic reactions. Antibiotic resistance is an important issue in modern medicine. Penicillin was the first antibiotic that was discovered, and that was discovered about 75, 80 years ago. And since that time, penicillin is now rarely prescribed because most bacteria have become resistant to it. And this doesn't just happen with penicillin, this happens with all antibiotics. This image summarizes the different ways that a bacterium can be resistant to an antibiotic. In other words, these are the different ways or mechanisms that bacteria can avoid being affected by different antibiotics. The first mechanism of resistance is for the bacterium to prevent entry of the antibiotic into the cell. So this is also described as decreased uptake. And this could be due to having the production of different protein transport molecules. So some antibiotics are allowed passage into the cell by different protein transport molecules. And if those molecules change shape, that will prevent the antibiotic from entering into the cell and having its effect inside the cell. The second mechanism of resistance is pumping the antibiotic out. So in this case, the antibiotic is able to enter the cell, but it is immediately pumped out. And this is called efflux. So where that antibiotic is pumped out by protein pumps. Third mechanism of resistance is to produce inactivating enzymes. So the bacteria may produce inactivating enzymes, and these enzymes will break down the antibiotic. And this can occur outside of the cell, or it can occur inside of the cell. But either way, the antibiotic is altered so that it can no longer function. The fourth method of resistance to antibiotics is to use an alternate enzyme. So if the antibiotic has an effect on a particular enzyme and inactivates the enzyme, then the bacteria can use an alternate pathway to produce that end product that it needs. So in this case, the antibiotic is still functional, but the bacteria can use an alternate biochemical pathway to get to the end product that is desired. The last method of antibiotic resistance is to alter the target protein. So in this case, the antibiotic may recognize the ribosome or maybe the RNA polymerase that is involved in transcription. If these proteins are altered in a way that the antibiotic can no longer recognize the protein, then the antibiotic will not be able to affect the activity of those enzymes. So these are the 5 mechanisms of antibiotic resistance. First, altering transport proteins, or producing efflux protein pumps, or producing inactivating enzymes, or using an alternate enzyme, or altering the protein products. Now, one of the things you should notice about all of these mechanisms of antibiotic resistance is most of them involve some sort of protein. Decreased uptake involves protein transport molecules. Efflux pumps, protein pumps, inactivating enzymes producing enzymes, alternate enzymes using a different enzyme, and then altering the target, altering these proteins, these enzymes. In a previous lecture, I went through the process of gene expression. And just as a reminder, a gene is a sequence of DNA that encodes for a protein product. And the process of gene expression begins with the DNA, which is copied into an mRNA, messenger RNA. And that process is called transcription. Then that mRNA is used to produce a protein product. And that process is called translation. So genetic change in bacteria is important because if a gene is altered, that can lead to a new protein product, or if the bacteria acquire a completely new piece of DNA, a new gene, that will also lead to a new protein that can be produced for that bacteria. And the importance of this relates to antibiotic resistance. So if a bacterium acquires a new gene or acquires a mutation that produces a changed protein product, that can lead to the bacteria being able to produce a protein that makes them antibiotic resistant. So if these bacteria were able to produce some sort of protein, like an inactivating enzyme, they are now resistant to that antibiotic. Also in the genetics lecture, I went through the different methods by which bacteria can acquire new genetic material. Mutation is one way bacteria can acquire new genetic material. And in one of the previous lectures, when I talked about genetic change, I went over this slide with mutation. So in the slide, the top portion shows the wild type, which is the normal DNA sequence, and then the normal protein product. A mutation occurs when there is a change in the sequence of DNA, which leads to a change in the mRNA, which could possibly change the protein product. And in this case, it is a missense mutation where one amino acid has been altered. Now, the benefit for the bacteria in this case is that if there is a mutation that slightly alters the protein product, where the protein can still be functional, but the structure has been slightly altered, this could result in changing the target for an antibiotic. So for instance, if this is the gene for the RNA polymerase, and there is a mutation which slightly changes the structure for the RNA polymerase, but it is still functional, then it is possible that rifampin, which is the antibiotic that can recognize RNA polymerase and inactivate it, it's possible that now the rifampin can no longer recognize the RNA polymerase, and now this mutation has caused the bacterium to become resistant to rifampin. Another method by which bacteria can acquire new genetic material is by transformation. And transformation occurs when a bacterium can pick up naked DNA from the environment. So the bacterium will pick up the naked DNA. The naked DNA can then splice into the chromosome, and now whatever gene was on that segment of DNA, that bacterium can now produce the protein product. So for example, if there was a gene there for a protein pump, that can pump out the antibiotic before it has its effect. Now this bacterium has acquired that ability to perform efflux, to pump out the antibiotic before it has its negative effect. So now this bacterium is resistant to that particular antibiotic. Transduction is another method by which bacteria can acquire new genetic material. And the basic definition of transduction is where bacterial genes are passed from one bacterium to another by a phage. And there are two types of transduction. There is generalized transduction and specialized transduction. Generalized transduction is associated mainly with lytic phages, and during the life cycle, during penetration, the host chromosome is degraded, And then during maturation, some of that host DNA is accidentally packaged into the capsid. So when that phage particle infects a recipient cell, it is penetrating or injecting bacterial DNA. So that bacterial DNA will recombine with the bacteria's chromosome, and now whatever gene was packaged into that phage particle, the cell now has the ability to make the protein. Specialized transduction is associated with lysogenic phages. Remember, lysogenic phages can form a prophage. So the prophage is that spliced phage DNA, and it splices into the bacterial chromosome. When the prophage gets the trigger to splice out, it accidentally takes a bacterial gene with it, so it doesn't do a perfect splicing out. Then, as it goes through biosynthesis and maturation, all of the phage particles will carry a copy of that bacterial gene. So when those phage particles infect a recipient cell, so when they inject their DNA into the recipient and that DNA splices into the host chromosome, forming a prophage, it has also brought with it a bacterial gene. And if in either of those cases, the bacterial gene is a gene for an enzyme that can break down an antibiotic, then these recipient bacteria have now become antibiotic resistant because they are able to now produce an enzyme to break down that particular antibiotic. Probably the most common method by which bacteria acquire new genes is by conjugation. And conjugation involves a F-positive cell that has a plasmid and has the ability to form a pilus to an F-negative cell. And what the F-positive cell will do is it will transmit a copy of that plasmid across the pilus to the F-negative cell, converting that F-negative cell into an F-positive cell. And whatever genes are on that plasmid, those will be genes that will now be available to this recipient cell and will give that recipient cell new characteristics. There are many different types of antibiotic-resistant genes that are found on plasmids. So this is an example of a plasmid. This is an antibiotic resistance gene, and in this case, it shows that the gene encodes the information to make an inactivating enzyme, an enzyme that can break down the antibiotic and make it non-functional. But there are plasmids that also contain enzymes that can modify antibiotics, or they can produce a new type of enzyme that has an altered target site, so now the antibiotic can't bind. There are also genes on plasmids that encode efflux pumps that will be able to pump the antibiotic out, or there are genes on plasmids that encode the information to make porins that prevent entry of the antibiotic. So there are many different types of antibiotic resistance genes that are found on various plasmids. In order to really understand how a population of bacteria can become antibiotic resistant, it's important to understand the process of natural selection. You've probably heard about natural selection in a basic biology class as the mechanism of evolution, which it is, but it also explains how a population can become antibiotic resistant. So the basic process involved in natural selection is first to start with a population of individuals, and there needs to be variation in the population. In other words, they're not all identical. Then in their environment, there needs to be a selective pressure. And what that selective pressure means is that not all of the organisms, not all of the individuals will be able to survive, but some will survive. And those survivors are going to reproduce. And because they reproduce, their genes are going to be passed on to the next generation. And over time, if this selective pressure remains in the environment, over time, the population's traits are going to change. Usually when natural selection is taught in a basic biology class, the examples that are used involve animals. So as a review, I'm going to use an example with animals. So let's say the environment you have is a valley. And in this valley, you have a population of rabbits. Now the important thing for natural selection is that these rabbits are not identical to each other. So you need variation in the population. So you can have variation in color. You can have variation in size. You can maybe have variation in how well they hear or how fast they run. You could even have variation in how well they metabolize glucose. But the point is there's variation in the population. They are not all identical. Now for natural selection to occur, you need a selective pressure. So in this example, the selective pressure will be the fox. So the fox has moved into the valley, and of course the fox is going to hunt the rabbits. In this case, there will be certain rabbits that will be more likely to be caught by the fox. And the example we could use is maybe coloration. So these white rabbits are going to be more visible. So the white rabbits are more likely to be caught by the fox and not survive. So the rabbits that have the more camouflage type coloring are more likely to survive in this case. So that is the selective pressure, and that will allow these rabbits to survive that selective pressure. And then after they survive, or while they're surviving, they're going to reproduce, and all of their offspring will tend to have the same traits, the same coloration. And over time, if you look at the population of rabbits over time, with this selective pressure, the rabbits will have a darker coloration. And that is natural selection. So natural selection is where a selective pressure is only going to allow certain individuals that have advantageous traits to survive. So those traits will be passed down through the generations. Natural selection and human behavior can be used to explain why populations of bacteria become antibiotic resistant. And so an example for this could be a person who has strep throat. Let's say a person has strep throat, this is their throat, and in their throat they have a population of Streptococcus pyogenes, which is the bacteria that causes strep throat. And these bacteria are not all identical to each other. They have small variations. They can have variations in how they metabolize, and maybe small variations in the amount of peptidoglycan they have in their cell walls. All of them have a lot, but you could have small variations. But another variation could be how sensitive they are to a particular antibiotic. So that's the antibiotic. So this person goes to the doctor, gets a prescription for this particular antibiotic, and then they start taking the antibiotic. And most of these bacteria are fairly sensitive, so the antibiotic will start killing the bacteria. But what can often happen, and this relates to human behavior, is that often the person will not take the full course of the antibiotic because they start feeling better, since most of the population of the bacteria have been killed, and so they either forget or they decide to stop taking any more of the antibiotic. Well, what that has done is that antibiotic was a selective pressure. It killed off some of the bacteria, but you still have some survivors. So over time, what those survivors will do is they will replicate. And these were bacteria that were a little bit more resistant than the previous bacteria. So now these bacteria are more resistant. So when the person goes back and takes the same antibiotic, it won't be able to kill as many of the bacteria because now these are more resistant. And over time and exposure to antibiotics, eventually the population of bacteria will become completely resistant to the antibiotic. And when that person is exposed or takes that antibiotic, it won't work at all. So it won't kill any of the bacteria. So this is an example of natural selection. Natural selection, you have a population of microbes, a population of bacteria. They are exposed to a selective pressure, the antibiotic, and that antibiotic will kill off the sensitive bacteria, but then those that are resistant will survive and replicate, and eventually, over time, over being exposed to the same antibiotic over and over, you will eventually develop a resistant population. Most of this chapter was over antibiotics, and there are many different types of antibiotics, hundreds of antibiotics that are selectively toxic. Again, selectively toxic means that they affect the bacteria and not the eukaryotic cells, not the human cells. There are other antimicrobials that target other types of microbes, not bacteria, but there are many fewer options for the other antimicrobials. And keep in mind that the whole purpose of the drugs are to be selectively toxic. There are very few options for selectively toxic antiviral drugs. And this makes sense if you remember viral replication. Remember that viruses are essentially a capsid protecting a nucleic acid, and that in order to replicate, the viruses have to go into the host cell, into the human cell, and use the machinery of the human cell to make more copies of it. So to stop virus replication, we have to stop normal human cell function. And so that's very difficult to find selectively toxic drugs, because essentially to stop virus replication, you have to stop the human cells from functioning. There are a few antiviral drugs that are selectively toxic, and this is because they target enzymes that are associated with particular viruses. The first example is acyclovir. Acyclovir is associated with herpes infections, and herpes viruses are viruses that are DNA viruses, so that viral genome can splice into the host chromosome. Now, associated with a herpes infection is an enzyme called thymine kinase, and that is only found in cells that are infected with herpes. And what can happen is the acyclovir will cause that thymine kinase to produce a false nucleotide. And that false nucleotide will prevent DNA replication. And so that is associated that acyclovir only works for herpes infections. Another virus that actually has some selectively toxic antiviral drugs associated with it is HIV. Remember that HIV had that special enzyme, reverse transcriptase, that is not found in human cells. And reverse transcriptase takes the viral genome, which is in RNA form, and converts it to viral DNA. And that viral DNA then splices into the host cell's chromosome. Well, that's a perfect target for selectively toxic antiviral drugs. That reverse transcriptase is a common one, and there's also another HIV protein called integrase. So targeting enzymes that are only associated with the virus results in selectively toxic treatments. Fungal cells are eukaryotic cells, so their structure is very similar to animal cells, to human cells. So to find an antifungal drug that is selectively toxic, you need to know the differences, the structural differences between a fungal cell and a eukaryotic cell. And two of the major differences is that fungal cells have a cell wall, and the major compound in the cell wall is chitin. So some of these drugs affect or target the cell wall, and they are targeting the chitin. Another difference is the sterol that is found in the cell membrane of a fungal cell. In fungal cells, it's ergosterol. In animal cells, it's cholesterol. So most of the antifungal drugs are targeting either the cell wall or the ergosterol, and these are two structures or two compounds that are not found in human cells. When talking about antiprotozoal drugs, it's important to remember that protozoa are eukaryotic cells also. When we think about a human cell, this is a basic diagram of a typical human cell. When you think about a protozoan, this is an example of a protozoan cell. And if you look at the different organelles, there is a lot of similarity. They both have ribosomes, and these are ADS ribosomes. Protozoa also have mitochondria, they have an ER, they have a nucleus, a nucleolus, they have a cell membrane, they have a Golgi. So they are very, very similar to human cells, which means that most of the antiprotozoal drugs are not selectively toxic. So if a patient is taking an antiprotozoal drug, it's really important that the dose is correct, because if they take too much of the drug, it will eventually damage their bodies and can damage their cells. Helminths are the last group of microbes to talk about, and the helminths are the worms, and that includes the trematodes, the cestodes, and the nematodes. And the important thing to remember about the helminths is that they're not just eukaryotic organisms, they are classified as animals. They are multicellular and they have body systems. So if you look at the anatomy of this nematode, you can see that there is a digestive system. So it has a mouth, it has intestines, it has an anus, and they have nervous systems. So they have nervous systems, and they also have reproductive systems. So these organisms are the closest related to us in terms of how the cells function and the body functions. Most of the antihelminthic drugs are going to affect energy production by the helminth or paralysis. And all of these drugs are not selectively toxic. So in higher doses, they would be able to affect our ability to produce energy and also our nervous system. So with antihelminthic drugs, again, it's important to pay attention to the dose, because higher doses of these drugs could have a toxic effect on the human.